Let's continue. So the main schedule of the metabolomics between LCMS and GCMS is almost the same. So in the second part, we choose the type of samples for the experiment. Then we do the metabolite extraction. For liquid sample and solid sample, the SOP is different. But in the most case, mainly our target is to as more as we can to extract the metabolites out of sample. And after the extraction, we do the fingerprinting profiling. Mainly, there was 3 different parts. The first is to separate the metabolites. We use LCMS or GCMS, the chromatographies, to separate the entities. And then we have to shoot them to the mass analysis. But in the mass analysis, we have to do the normalization to make the metabolites with a different chart. And there are different types of methods for the ionization. For LC, we have the ESI or APCI methods. And for the GCMS, normally we use EI, the electron spread ionization methods for the ionization. And then we use a mass for detected. And there are different parts of mass, but in GC, normally we use GC TOF. But in LC, we can use a LC QTOF or LC Orbitrap, different kind of type of machines for quantification. These parts are totally called the fingerprinting profiling. After that, we have to do the data processing. And normally, the metabolites in biological samples had a very large concentration range from like pp2 to ppm, or even more than that concentration. So we have to do normalize and transformation to make the concentration at around a limited range of and then we have to do the data analysis like the PCA or PLSDA to select the statistical analysis compounds, which we can to verify them to like that. So then after the statistical analysis, we got some compounds. Maybe it's very important. Maybe just biological significant, non-meanings of biologicals, just a statistics analysis. So after that, we got the markers selected, and then we do the annotation. The annotation, normally there are 2 parts. The first, we have to know the element composition, and then we have to know the structure of it. So in most cases, the softwares will do it together, but actually there is 2 different parts. After that, we have to do the targets of metabolomics to do the validation. Normally, we have to buy the standards to validate the compounds identified is correct or not. But if we go to this part, the numbers must be not too many, not thousands, just several kind of metabolites. We have to validate it, and then we have to do the interpretation. But in GCMS, the schedule is a little bit different. Before the inject to the GCMS, like in this part, the part here, we have to do another step that is called, before this, we have to do the derivatization to make the metabolites, which can be detected in GCMS. So it's a little bit different. And as well as here, after the GCMS acquired the data, we do not need to do the statistics directly, or we have to do the annotation first, and then we have to do the statistics analysis. This is a little bit difference between the GCMS and LCMS metabolomics. But anyway, the metabolites is different. For GCMS, for metabolomic study, we have to do the TMS derivatization. This step is pretty necessary for the GCMS because not all the metabolites can be turned to gas at a certain temperature. So when we do the TMS derivatization, the metabolites, the gas point, will turn lower and lower. It could be easily detected in GCMS, as well as the polarity will get stronger, not that lower. So so we can see that what kind of metabolites can be detected. So if the metabolite needed to be TMS derivatized, it should have the functional engine like the hydrobones, like the carboxyls, or the mercaptose, like these kind, or amines. These kind of functional engines could be derivatized by TMS. So let's see that in the HMDB library, we have around like more than 100,000 metabolites, but only around 70,000 metabolites could be TMS derivatized. So which means more than half of them cannot be derivatized. So it cannot be detected in Gcms. But in the fact, more of them, the molecule is too big, even after derivatization, and it cannot be easily turned to gas. So in this ways, the metabolites detected will be much smaller and smaller. Let's see that the normal GCMS database, you will see that, let's see the ontologies of these metabolites. Normally, they are amino acids, organic acids, sugars, pentones, alcohols, fatty acids, and cholesterols, and their derivatized compounds. So these metabolites are not cover all the global metabolomics pathways. So the first thing that the detected metabolites in GCMS is restricted. It's just only primary metabolites with molecular weight between 70 to 500. So the background is the global pathways. I downloaded from the 𝐾𝐜 database. You will see that the GC could only detect the red point. So it's not that too much. When we see that normally in my lab we use a database contains like 1,640 unique metabolites. It was combined with different library together, like mainly from the 𝐕 lib, the Oliver 𝐕 lab, so and the 𝐉𝐭 lib, and also some metabolites we detected in my lab, around 70 compounds. So it's not too many. Like there is a study published in UC Davis, by UC Davis, West Coast Metabolomic Center. You will see that they detect the metabolites, use different platforms, LCMS combined with GCMS in the mouse, liver, brain, and blood. Totally, they identified around a little bit over than 1,000 metabolites, and most of them are lipids. So they use LCMS for lipidomic study. In the GC-TOF, the primary metabolites is around 200 metabolites could be annotated. So which means that the metabolites not too many. So in the GCMS, the identified metabolites normally range from 150 to 400 in currently technologies. Another important thing that when we do the metabolomics in GCMS, the first step, that is to extraction. Then we have to do two important steps. The first step is to methoxymation. This step is to methoxylize the pure ketones and aldehydes reaction, not all the carbonates. Like the ketones here, we can do the methoxymated and to get two different shapes of byproducts. And after that, we're going to use a TMS for derivatization. But this step will generate lots of isomers. Like this one, we have the keto position, the alpha keto here, and also we have the aldehydes here. And after the mass oxidation, you will get to 4 different isomers, which means these compounds will be generated 4 different isomers. And look at this case. There is a compound here. You will see somebody say that the ketones here or the aldehydes here will be mass oxylated to different shapes. Actually, it's not, because they are kind of carbonyls. So the carbonyls will not react it. Only the two ketones and anhydrous will be reacted. So one compound will be generated two or more than different isomers. And in the derivatized part, the TMS derivatized part, you will generate more multiple compounds. Like the aniline here, it has the carbonyl engine here and amines amino engine here. So the carbonyls will connect with one TMS, and the aminos will connect with like 1 or 2 or 0. So they will get several different possibilities. So the byproduct is a lot. When we check the database, like how many alanine was there, you will see that there were more than 3 different kind of the EI spectrum there at different retention time. So this is one point that there is a case, like the most complicated thing, that is the glucose in GCMS detection. For GCMS, the glucose will be methoxymated to two different isomers, and then later for silylated, you will get two different peaks. One we call major, the another called minor. Their actually their structure, the EI spectrum is almost the same. Let's see that the glucose with 5 TMS, the major shape and the minor shape, you will see that the spectrum is totally the same. So if we only use EI spectrum similarity for and identify the metabolites, you will get wrong. Another thing that there is some kind of SV, lots of the molecules with the same illusions in GCMS. Like here, there is a D-shaped glucuronic acid. The major shape was exactly the same point, same retention time with sorbitol, the D-shaped sorbitol. So in this case, when we have to identify or quantify the metabolites of the glucuronic acid, we have to use the minor ones for quantification. If we use the major ones, there will got something wrong, or where it will be affected by the concentration of the sorbitols. Especially the sorbitol concentration is much higher than the glucuronic acid. So let's get a comparison. You will see that in GCMS, because of the step of derivatization, you will get lots of byproducts. But these byproducts can be detected at a different retention time. Like the glucose here, in major and minor shape, there were two peaks because of different shapes after meth oxidation. But in the LCMS, they also have lots of the various compounds, but they are in the same retention time, just with different adductive ions, like in the hydrogens or the sodiums, some kind of adductive ions were combined with the molecule. So these are two differences. So the derivatization will produce many byproducts in GCMS. If we detect around 1,000 featured there, actually there's less than 500 true metabolites are there, because many of them are produced by derivatization. Okay, let's see. Another thing that difference from LCMS and GCMS is that the GCMS must have the steps for deconvolution steps, because the totally difference, the principles of GCMS and LCMS. We use the EI ionization methods. It means that at one point, many metabolites are ionized together and fragments in the source. So it will get all of them in the mass, and they will released from the columns in the same point, some of metabolites. So like here, we have 3 metabolites or 4 metabolites. They are released at a very close time. If we double click here, you will got a peak of fragments from 4 or 3 metabolites, not from 1. So this spectrum is not useful. We have to do the deconvolution to separate them to 1, 2, 3, three different spectrums from three different compounds. Here is this example. Let's see that if I double click the time point between 6.3, 6 to 6.68, you will get a spectrum. We call it the raw EI spectrum. If we use the spectrum to against the library, you will get nothing, or you will get the wrong result. So we have to use the algorithms to do the deconvolution, to remove the spectrums or the peaks not from these compounds. And then we will reorganize all the peaks together to get a computed the deconvoluted EI spectrum here. Let's compare these two spectrums. You will see that the spectrum is different. So then we can use this one to against the library. So which means the EI spectrums, it was reorganized by algorithms. And also there is something may happen if the algorithm is not good. You can see here in the samples, we got compounds called caprylic acid. Actually, if we buy the standards for injection, you will get the spectrum like this. And softwares after deconvolution, they will give you the spectrum in this one. So let's see, compare the spectrum 1 and spectrum 2. The second is the standard. The first one is the metabolites in the biological samples, deconvoluted by software. So you will see that many peaks was not the same. Actually, they are contaminated by the matrix or by another compounds, and it cannot be separated. It cannot be calculated, deconvoluted by the algorithms. So in this case, you can easily get wrong. Maybe you choose the ions that was contaminated ions for quantification. You will get the wrong result. So which means that Gcms need to be deconvoluted, and the EI spectrum was affected by the algorithm. If the algorithm is not good, you cannot get the pure EI spectrums. It will leading to the wrong results. Anyway, Gcms or LCMS better. We cannot say this because they both have their disadvantages or advantages. In the Gcms, the column is pretty long. Normally, it's longer than like 30 meters. It's a pretty long column, so the separation is pretty good. Like it can be easy to separate the fatty acids, like the 18 carbons with one unsaturated carbons. So these 6 isomers can be separated in Gcms, but in LCMS they cannot be separated. They just one peak. Also in the sugars. In sugars, as we know that if we use the LCMS, the separation is not good, especially for normal columns. If you use a specific columns for sugar, there will be much better. But in GC, we can see that the sugars can be separated very good, very good. So examples here, like there is 6 different sugars. In the Gcms, we can separate them very good, but in the LCMS, it cannot be. So the separation GC is much better than LC. But in LC, it has the precursor ions. In GC, it cannot have. So because we use the EI ionization, there cannot be generate a precursor ion, or the precursor ion intensity is pretty lower. So normally we ignore it. But in LCMS, we use this for quantification. So let's talk about some annotations, pitfalls. In the annotation, normally we have 2 strategies. The first strategy is to do the similarity against the library and the retention time calculation compare with the library. So we do the annotation. The second thing is we use fermentations to do the structure elucidation. So when we got the spectrums, we use the element like formula prediction based on to calculate what kind of element they're gonna have for this. But in this ways, we have to know the precursor ion. If we don't know the precursor ion, we cannot do this. And then we use softwares help us to do the fermentations to see each fragments, what's the structure of it, and then to combine them together, to combine them together to get a molecular shape. But there is some neg false tip here. So in Gcms, normally we use spectrum similarity combined with retention informations for annotation the metabolites. In LCMS, we have the precursor ions and spectrum similarities, as well as some case we can use the retention time or the isotope pattern. If we have the ion mobility tube, we can calculate the CCS value for annotation. Normally we use the first two to annotate it, the metabolites. Like here, let's see that in the LCMS, the one compound, if we use different collision energy, it will get different spectrums. If we compare them one by one against another one, you will see the similarity is not that good. It's less than like 0.7 or something. Like here, if we use different kind of LCMS for metabolomics, we have to take into consideration that the database we used must be matched with our machine. Like this one, if we use the orbit ion trap 𝐾 to detect it, or the 𝐓𝐢𝐭𝐿 CID collision fermentation to do the MS acquisition, if we compare them, the spectrum is not the same. Actually, they are different, but it's the same compound. All is LCMS. Just because one is orbit trap, the other is the TOF. So their spectrum cannot be compare each other's. But this is an extremely example. In most cases, they are the same, but there are some difference. So we have to be considerate. But in the Gcms, we use a collision energy like 70 eV. It's pretty good. All the machines we use the same collision energy, which means that if we use different machines for detected or to acquire the EI spectrums from the same compounds, like the 𝐒𝐞𝐿 TOF, Angelan 𝐺𝐶_q_mass, Shimazo 𝐺𝐶_q_mass, their spectrums is almost the same, which means that the Gcms can be shared the libraries. The database can be used in different machines. So it's pretty good. But another thing that is we have to know that the EI spectrum is not enough. We need another information. Like here, we have the lactitose spectrum here, and we against the library, we will get this one, the maltitose. Actually, the lactitose and maltitose are isomers, so their spectrum are almost the same, with pretty good dot products value like 0.99. And if we see that the second method is beta cell ribose, the beta cell ribose is different from the lactitose. Like here, it's kind of a chain, and in the beta cell ribose, it's kind of a ring. So the structure is different. Structure is totally different, but the spectrum is pretty similar. The dot product results 0.97. As we know that when we do against the library, we normally set the dot product tolerance to 0.7. So 0.9 is a pretty higher similarity in this case. So if we do not to take the information of retention time into consideration, we will say that it's a very good match. You will see the third match, the gentile bias. The structure also different from the lactitose, but the EI spectrum is also pretty similar with 0.98 dot product value. So it's also very good. Let's see the fourth one. The fourth one is sucrose. Sucrose is another sugar, but the structure is also different. It's also a ring here. It's a chain here. It's different, but similarity is good. So this is the fourth one. And the fifth candidate has 3 different sugar rings combined together. the structure, we can see that they are not the same, almost different compounds. Actually, they're different compounds, but let's see the spectrums. The EI spectrums also can be matched together with 0.9 dot product value. So which means that if we use only the EI spectrums for annotated metabolites, we will get wrong. Let's see what were difference of them. Their retention index is different. So the candidates retention index, is ranged from like 900, around 900,000 to like 1 million. So the retention index is different. When we use GCMS for metabolomic studies, the first important thing that is we have to use the retention index. If we don't use it, we will get the wrong annotation. So how to use it? We normally use the fatty acid methyl ester, like this ones, to calculate the retention index of each metabolite, and then compare them with the library. In most cases, the library contains the retention index information. Like here, if we do not use the retention index, you will get the compounds, like each dot is one compound here, and we have the experiment retention time, and the metabolite annotated, it has a retention index in the library. If we do the linear, you will see that lot point was not linear, and these metabolites are annotated wrongly. So if we only cut the EI spectrum cut off at 0.7, we got 242 metabolites in this case. If we added the retention index like smaller than 500,000, it will get 185 metabolites, which means the gray metabolites will be removed. Only the red one will be kept. So the retention index is pretty important in the annotation part. If we do not use it, you cannot annotate your metabolites rightly. Another thing is quant mass. As we know that in the final data set for metabolomics, we have each metabolite annotated rightly in samples, as well as we need to know the intensity of each metabolite. But intensity, if we use it, we have to know which ions used to quantify it. We call it quant mass. In the LC-MS, we normally use precursor ions, the molecular ions, for quantify these metabolites. But in GC-EI-MS, we do not have the precursor ion. All the precursor ions is pretty lower. So we have to need the fragment ions to quantify the metabolites. Let's see that in the library. You will see many metabolites were used 147, 117, 103 at the quantum mass. Like this one, 147. There is over 300 metabolites. Use this one to quantify the metabolites. And the software is not that smart. It always makes mistakes. Let's see. Like here, we use the MS style for the data processing. Like here for extraction and identification. The MS style choose the 147 as the quantum mass for most metabolites here. You can see there is a line here. It's almost matched with the database. But something will happen there. Let's see. Let's see, these metabolites at different retention index choose 147 as a quantum mass in the GCMS database. And here, if we do a real case, you will see if you extract as 147 as extract ions in the extract ion chromatography, in the serine sample, you will get extract EIC figures like this. And if you zoom in a random position like the 6.6 here, you will see there is 3 different metabolites with all of them have the 147, and their retention time is pretty close. So when we have a large samples for quantification, the software will make a mistake. Will it happen? Actually, it's really happen. You will see that we have different samples around like 100 samples here. And if we are doing the alignment, you will see the software make mistakes. There is 3 different metabolites here. The peak one is kind of a sugar, and peak 2 is isotope of this sugar. And the peak 3 is 2-oxyglutarate acid, kind of a metabolite like this. But all of them has a peak 147, and this is the main fragment peak of these 3 metabolites, and they are pretty close. Softwares were annotated wrongly. Actually, I want to quantify the sugars like in the D-threonate acid sugar here, but most of them was quantified here, and some even quantified in the 4th peak. So the software did the wrong calculation. It had to be manually checked. If we do not do this, actually the consensus is not this metabolite. The peaks was choose the 2-oxyglutarate acid, so it will make mistakes. Actually, there is some other thing that is the contaminate from others. I said that the deconvolution algorithm is not that smart. It cannot always give you the pure peak. Sometimes it gives you the peaks from cannot be separated by 2 compounds with 2 compounds. Like here, the uric acid always has some contaminated ions from the insoluble compounds. Some compounds was from this one, so the spectrum is not pure. This case, I think we have to manually check it or manually to figure it out. So let's see that the compound 147 was contaminated by its neighbor. Actually, its neighbor. Their retention index is pretty close. It's pretty close, almost the same. It's under the tolerance, so it will be affected. Finally, we're going to talk about the libraries here. Actually, in this library, we have to show them before, but you will see that in the LCMS, normally we will detect like 5,000 fixtures, but in positive mode, and around 4,000 fixtures in negative mode. But it's different by the difference in metrics. But the library, like we use the library in the MONA database, we will get around 100,000 metabolites there, spectrum there. And in the negative mode, we got around 50,000 featured the LCMS spectrum here. So the library is much bigger or even huge than the spectrum we detected. But in the GCMS, it totally the opposite ways. In GCMS, we normally detect less than 5,000 feature there. But in the library, it's pretty small. Like the most important, famous, the FinLib library, it contains only 1,180 spectrums in the library, but we can detect around 5,000 features there. So in this ways, the library is not enough. We have even to see how many unique metabolites there. It's around 700 spectrums in the library. Actually, this is a very important library in the GCMS metabolomic studies. We use it. So in this case, many metabolites cannot be annotated in GCMS. So most of them are unknowns. So does unknowns important? If the library contains only known metabolites, maybe you annotated wrong. In this case, we have a salmonella samples. We use a GC-top to detect it, and we got around 2,078 featured here, and we annotated with the library. This is the library we used in our lab, and it only has metabolites which has names there. So with unknown beings, we set the tolerance, the EI spectrum similarity larger than 0.7 and retention index lower than 10,000. We will annotate 192 metabolites. But if we use the library with the unknown beings, which means that most of them are unknowns, only 1,074 metabolites with names, and the others do not have names. It's just the spectrums, nobody knows it. So this library, we set the same parameters. We will got around 420 metabolites. And the 420 metabolites has 132 metabolites with name and 282 metabolites with unknown beings, which means they do not have name. They only have the spectrum and retention index information. So it annotated with just a number, a library number. So if we do the against with the unknown beings with 182 metabolites with names, you will see that it has 69 metabolites, which was overlapped with unknown beings, which means it could be annotated as unknown beings, but we don't have it, so it annotated as a second candidate. So let's see what was happened there. So let's see what was happened with the 96 metabolites overlapped with unknown beings. Like this one, if we use metabolites with unknowns, we will calculate it against the library. We got this one. This is called the cadaverian, and this one we have the retention dot product as like 80, reverse dot product as 80 to 84, and the delta retention index like 4,300 or something like that. So which means that all of them are lower than the tolerance of similarity parameters we set. So we say that this is correct. But we use unknown beings. There is another one with matched, and this is matched much better. The similarity is a little bit better than the ones we used, and the delta retention index is much lower than that we used in the in another library. But this one do not have the name. It has the 3,307, and this one, if we check it, many samples can be detected. This one, but we don't know its name. Especially for this one, it's kind of a microbe or something like the Marinus. Marinus is in the marine. So this one may be annotated lonely, but we cannot know it if we do not have the unknown beings. Another case here, this one, when we annotate it, they give me the result like N-acetyl beta D-glucosame. It's very normal compounds, and the delta retention index like 2,000, and dot product and reverse dot product is very good. It's over than 90. It's pretty good annotation results. So in most cases, we are pretty confident with the result here. But you will see there is some difference, especially for the 103 and 147. The two peaks percentage is different. In the library, it's almost close, but in the real samples, it's a little bit different. But if we detect it in the unknown beings library, you will see it give another candidates. This candidates also has a pretty good reverse dot product and dot product results, as well it has a delta retention index like 400. It's pretty low, almost the same. And this case, you will see that the 103 and 147, the percentage in the library and the experiment is close. So I was thinking maybe it was annotated wrongly. If we do not have the standards, maybe it's wrong. So let's check what this being ID 1684 detected. In most cases, it can be detected in B or in some other plants. So actually it exists, but just we do not know it. But if the library do not have it, we will annotate it wrongly. So what was the future of GCMS metabolomic study? I think the first thing that is the database. We need the EI spectrum database with much more metabolites, even they are in silico. So if we do not it, we cannot detect more metabolites or annotate more metabolites in GCMS. The another thing that we need to detect the GcQQQ based EI broad target metabolomic technologies. That means that we're gonna have many standards to build this library. But if we detect it, it will much be useful. The third thing that is the high resolution CI sources GCMS technologies for metabolomic study. As we know that when we use the EI sources for the GCMS metabolomic studies, we never talk about resolutions because the resolution is pretty lower. We even do not need to see the values up the dot, just before the dot, which means that the resolution of EI sources GCMS is pretty lower resolution. So if we have CI sources like high resolution database, I think that will be much better. The fourth thing that is application of the 2 dimensions GCMS data metabolomic study. Actually, they has been used these days, especially for the volatile studies. We use 2 different columns, like we call it 2 dimensions. So we can separate the metabolites much better than only 1 dimensions. And then you will get a 3D like TIC figures, much better. Okay, finally, let me introduce the research team of our lab. So we are cooperated with 4 different labs together. One is the UC Davis Georgia 1 lab, and the Purdue University Brian lab, and UC Davis Finn lab, and also the Jiangnan University Shun lab. And the 4 labs were combined to do the researches. And this is the photos of the. Actually, this is my university, the Jiangnan University, the South Gate, the South Gate. It's pretty traditional gate, but it's very beautiful. And okay, this is what I want to show you. So welcome, you guys could have chance to Jiangnan University for visiting. Thank you.
Thank you very much, Dr. Jian, for your very comprehensive and I could say very insightful lecture on GCMS-based metabolomics. Indeed, I learned a lot in your lecture. So
Thank you.
now actually is the Q&A session. Please, if you have any questions, the audience, you can type in the chat box and I will read it for you. But yeah, we have, before I ask my question, because we have one question from Dr. Tan Jan Kit from UKM. So very informative talk. The question is, if we have limited budget and can only choose one platform, which platform, LC or GC, do you recommend for detecting human primary polar metabolites in an untargeted approach, without considering dietary carbohydrate, which GC it outperform LC as you mentioned?
Okay. Well, I would like to say that the platform different will generate different results. Normally, I want to know that what's the purpose you want to do this experiment. If you want to find the biomarkers of some disease or something like that, I think the LCMS is much better because it gives you the possibilities to discover new metabolites or normal metabolites. But in the GCMS, you cannot find the new one because it's not easy to do the to find the new one in the primary metabolites. It's just primary. But if you just want to see what kind of pathways was affected or do the metabolic profiling, I think GCMS is much cheaper. The machine is cheap, and also you just need to inject it. But in the LCMS, you have to inject it two, in negative mode and positive mode. And in most cases, LCMS need much more. You have to maintain the machine's performance because it's high resolution. But in GCMS, you don't need to do this because GCMS is not easy to, it's easy to maintain. So I think this case just depends on what kind of purpose you're going to do. In my experiments, normally we use the MTB and maxinol and water for extraction. So we can extract the polar metabolites and non-polar metabolites together in different two tubes, in one tubes. So we separate them in the tubes, and the polar ones, we're going to shoot them to GCMS platforms. And for the lipids, we use the LCMS for detected. So in this case, we combine the result together. We normally get close to 1,000 metabolites in one sample. So the second question, that is without considering the dairy carbohydrates, which GC is outperformance LC, as you mentioned. So what's the question means? Without?
I think with we excluding the carbohydrate, because we know that GC is very powerful machine to detect compared to LC. I think, but I think you answered his question.
Okay. Yes.
Yeah. Okay, I have a quite basics question to start with. So for the GCMS, do you have for the extraction, right? Because based on the extraction method, then we will affect the metabolite that will be detected in GCMS. Do you have any suggestion what solvent that we could use and what we should avoid?
Yes. Actually, this is pretty important things. If we choose the wrong solvents for extraction, you will get maybe opposite result, actually. So normally we use the SOP from the lab of Oliver Finn, because I was worked there 1 year in 2016 to 2017. So we use a standard procedure there, and they publish it in the Metabolomic workbench. If we search the Metabolomic workbench, you will see a lot of the procedure there, and they use it and published in many papers. I think it's solid, and we do not do any modification because it's really not necessary. But if you are working on the methods developing, like you want to optimize a new method or optimize the beta recoveries of metabolites, I think you have to do the experiment, or there is no necessary to do it because many people have done this before. So I suggest to use the SOP from the Metabolomic workbench.
Right. There are, I think, question again from Tanjan Kit. Could you elaborate a bit about the targeted workflow that employed in your lab for validating the results from untargeted approach?
Okay. Yeah, the validation is pretty important things. As we say that in very high possibility that you will annotate it wrongly, and you will tell the wrong stories, the biological stories.
Yeah.
It's just because you annotated not correct. So like if you are a company, or if the lab is established for a pretty long time, maybe you have lots of standards in your laboratory. You can shoot it and compare it with library, with spectrum very detailedly. But if we do not it, you can just lie if you can only depend or based on the public library for detection, for the annotation. But you have to know that like in around 5 to 10 metabolites is annotated lonely. So if the untargeted metabolomic result, you have to use it as a base or for another experiment. Or I would like to say, the new experiment was established based on the untargeted metabolomic result. You have to buy the standards to validate that the result is solid and it's a reliable, and then you can do another result. But you just want to say that the metabolites, the untargeted metabolomic studies will give some statistic difference metabolites to do the pathway enrichment to tell a simple stories. I think it's enough. Maybe there is some metabolites was annotated lonely, but most of them is correct. The pathways will be not that much difference. So especially for the biomarkers studies, I think the standards purchase it or you have to synthesize it to validate it is very important. Okay.
The next question is from Sarah Mahmood. Which is better in determining the effective compounds in plant extract, GCMS or LCMS?
I think for plants detection, because there is lots of the actually the GCMS LMS is both of them is okay. I think the HPLC is much better, especially when to find the new like the new metabolites in plants, especially some ketones or some specific molecules with complex structures. Because GC is only for primary metabolite, like amino acids or some organic acids, pretty simple. For complicated ones, it was not in the database. The database will not contain this information. Or even I would like to say that in the GCMS, there is not a specific database for plants. So in this case, it cannot be detected, especially for the secondary metabolites by plants or something. The structure is complicated, and we cannot do the fermentations for EI structure elucidation for the EI spectrums. But for the LCMS structures, that will be much better.
All right, thank you, Dr. Janji. We go to the next question. Lots question coming up. What is the best analytical approach? Again, LC or GC for untargeted study for secondary metabolites from microbes?
Yeah, especially for the microbiome, the second metabolites. I think LCMS is much better.
LCMS is
it's
much
not
better.
primary metabolite. It's said it's second metabolites. GCMS can do it, but the coverage is not that big. Just very simple secondary metabolite. But and also it had to be included in the library. So I said the GCEI database need to be developed because normally it's not too big.
All right. We have question from Dr. Kamarul Azlan. Have 3 questions from him. The first question is signal to noise ratio important for GCMS deconvolution?
Exactly, it's very important. For the low intensity metabolites in GCMS, the deconvolution is not good. You can do the experiment to get a standard spark it in the real samples and to make a concentration differently or to dilute them differently. You will see that the spectrum, the EI spectrums, will get poorer if the concentration is pretty lower or close to the noise. So it will affect it a lot.
The second question, any software recommendation for GCMS deconvolution?
Yeah, if you only to do the GCMS deconvolution, the AMDIS is the very critical ones. But if you want to do the peak extraction and the deconvolution and identification together, I think the MSDIAL is much better. It was developed by Hiroshi in Japan, the 𝐈𝐍𝐿 library.
𝐈𝐍𝐿 library.
Yes.
Okay. All right. The third question: how to reduce artifacts or byproduct due to the TMS derivatization?
Yeah, it's a very important question. That I think the artifacts can be removed very easy, but the byproducts cannot be, because when we do the TMS derivatization, it will be generate lots of products. But if we use the MSTFA as a solvent, it will be much better than BSTFA. It will generate the byproducts much lower. About the artifacts, I think the most easy thing that is to, before you to do the extraction, you can dilute your samples at different concentration and then extract it. After the data acquisition, you can make the standard curve lines for each peaks. If they are not in standard, if not in line, which means it's artifacts. So you can remove it. In most cases, in the 𝐕 library, the artifacts was labeled. So if it matched with artifacts, the result will tell you that it's not a metabolite, it's artifacts. You can just remove it. Okay.
Yeah. All right. We have question from the Facebook from Akil Fitri. Thanks, Dr. Jian for an insightful presentation. Would you suggest a comprehensive way to have a valid data when working on untargeted analysis? What need to filter? Because compared to targeted approach, it might be observable from the data since we target it. How about the untargeted?
Okay. So to validate the data, actually there is this is a part of the data processing part. So there is many approaches to do it. The first thing I think we have to do the QC normalization to remove the system errors or the extraction errors. So there is many steps there. So we extract it, deliverize it, and then use machine to detect it. It will generate, each step will generate system errors. So we use the QC, the quality control to do it, to remove the system error. This is the first step. The another step, then we have to confirm or to validate or curate the metabolites annotated. We can we cannot 100% lie to the machines or to the algorithms. We have to check it. As the example I gave it, maybe it was choose a different peaks for quantification, or maybe it was matched with the wrong metabolites in the library. So something always happens if we only use specific parameters and softwares for quantification and annotation. So it was happen there. If we use the is it called the targeted approach? It will be much better because we cannot annotate wrongly. So we have the retention time tolerance and so it will be much better. But it's important. The problem that is the methods cannot be applied broadly because the conditions in my lab and your lab is different. The methods in my lab is okay, but to yours is different. Even we use the same machine, the same solvent, the same parameters, even the column is also the same. It always some difference in retention time. Even in the same lab, there it will be changed with time. So I think target is good, but we have to spend a lot of time to keep the machine on a very good condition. then it will be works. Yeah.
Right. Okay. We have another question from Tan Jan Kit. For pharmaceutical-based targeted metabolomics, we are complying to the FDA bioanalytical method guidance. Is that the same we have to follow? The same guidance for targeted metabolomics in validation step for biomarker or mechanistic study. Often I don't see in the publication the incorporation of blank matrix heavy isotope label internal standard in validation step. Could you comment on this?
Yes, if the FDI bioanalysis methods guidance, that would be much better, especially for pharmaceuticals. But in the research part, not a company, I say research. Normally, we cannot use the targeted metabolomics to find the novel metabolites or specific normal biomarkers. It's very hard because in most cases, biomarkers is like not primary metabolites or a very easily metabolized in the pathways. Normally, specific ones, it was methylized or something. It was metabolized to a new one. It could be a biomarker. It's different from others. So in this case, I would say that if you use FDI analysis, bioanalysis method guidance, that will be much better. In the publication for the blank matrix is pretty important things. And actually in my lab, we use the blank samples to remove the fixtures, which is pretty close, or the is pretty close that intensity in blank samples. We set the tolerance to 5 for change. If it's lower than 5 for change, then that in the blank matrix, we will remove it. For the isotope label standards, I think it's for the quantification. If you want to detect a very accurate concentration of that compound, I think you have to use the heavy isotope labeled standards to do it. If you do not need to know the very accurate compound concentration, it's not necessary. And in most cases, many metabolites do not have. We cannot buy or cannot purchase the isotope labeled standards. And that is okay.
All right. Dr. Jian, I have a question because during your presentation, you mentioned about the database for GCMS is much more lower than the features that we detected. So basically, we will have many unknown peaks or features. So in your opinion, if we get this peak, what should we do next? I mean, for the future study.
Yep. Actually, we cooperate with the Fin Lab, and we got as we know that they have a Bing base, which was collected around 10 years of GCMS study cases in their library. And because it called they collected the data and they do the data processing and do the calculation to see how many different spectrums are there. I mean, the different spectrums is with different retention time and different fragments. So there is a kind of algorithms to say it's different or not different. So with the years goes, they collect around 20,000, around 40,000 fixtures, but only 1,000 fixtures has name. Most of them do not have name. So if you use this big database for annotation, you will annotate a lot of metabolites, but many of them do not have name. So the important currently we are working with the Oliver Fin the project, actually we are preparing that to annotate all the metabolites in the bean base with algorithms or machine learning methods. So if it was done, and I think there will be much more metabolites will be annotated. All these are restricted to the Gcms, TMS derivatized metabolites, because we have if we have the NIST library or some other libraries, it also has many compound there, more than 10,000 or 50,000, but most of them are not TMS derivatized. It's just a normal shape. It's volatile. We can say it's volatile. It can be detected directly in GCMS. Okay.
So meaning, from what you telling us, it is actually important when we run GCMS, if we can have a database that also have the unknown, features, then it's good. The bigger the database is better for the future, for the machine learning also. So that more, because why I say that, because in LCMS, as we know, we already have few in silico. From the fragmentation pattern, they can come up with the compounds, putative compounds. Hopefully in GCMS also we can do that, right?
Yeah, if we have the in silico fragmented database, that will be much better. Like it was dependent on how complex is the metabolites there. Especially for the lipids, there is a very successful in silico database called Lipid Blast. It's also developed in the Fin lab by Tobias King. And Lipid Blast, it was widely used in the lipidomic studies, and all the spectrums was computed by algorithms. So it was good. But for GC, I think there is no the in silico database right now. And maybe the algorithm is not good, or no many people to do it because of the not many people use GC these days, and most people choose to use LCMS.
Use LCMS, yes.
Yes.
Understand.
So did you use LCMS or GCMS in your laboratory?
It depends on the what objective that we want to answer. Of course, if we want to know the mechanism, we will go for primary metabolites. Of course, we will use GCMS compared to LCMS. But if now, like, we are working on secondary metabolites, normally I use LCMS compared to GCMS.
Okay.
All right. Okay, I think that's all for today. Thank you very much, Dr. Jian, for very insightful, very, I think everyone learned something about GCMS today. It's like having a comprehensive lecture, how to do GCMS metabolomics, what to consider, because I think you're telling about the experimental design, how to design the experiment when you use GCMS, what to consider, what will happen, so that we will take into consideration the data that we produce is actually not a false positive, sort of like that. Again, thank you very much, Dr. Jian. So before that, we have come to the end of the webinar. Again, I would like to thank Dr. Jian for the very interesting presentation.